All right, in this video, we're going to talk about the processing of eukaryotic messenger RNA. Specifically, we're going to talk a little bit about the 5' GCAP, the 3' poly A tail, and splicing. And all of this is coordinated by the RBP1 subunit of the RNA polymerase protein, and specifically the carboxy terminal domain of RBP1. So here what we have is, this is in yellow, this is RNA polymerase, and in green here, that's the carboxy terminal domain of RBP1. In gray, these are all of the general transcription factors, and so we're sitting here at the Tata box, so we're sitting here at the promoter. And depending upon the phosphorylation state of RBP1, the carboxyl terminal domain, will determine which set of proteins are bound to it. And the first set of proteins is going to be the capping enzymes. And so as soon as RNA emerges from that RNA polymerase, so that's in green here, the capping proteins are going to recognize the 5' end of that messenger RNA, and they're going to put a GCAP there. And we'll look at exactly what a GCAP is momentarily. And as the production of messenger RNA continues, then we have in blue here, we have the splicing factors. They're going to recognize introns, and they are going to splice them out and splice exons together. Now this happens as transcription is occurring. And so as soon as an intron is out, it's going to get spliced. As soon as an intron is produced, it's going to get spliced out. And finally, once the cleavage signal is transcribed into messenger RNA, that's going to be recognized by some enzymes that are involved in the production of the poly A tail and the cutting of RNA and the release of that RNA from the enzyme. So now we're going to look at each one of these in a little bit more detail. The 5' cap is here we have the 5' end, right? There's the 5' end of the RNA, so the beginning of the RNA that's being made. And the 5' cap here is a methylguanosine. So it's a nucleotide, but it's been modified. It's been methylated. And now it's added, instead of 3' to 5', it's added 5' to 5'. So we have this 5' phosphate, phosphate, phosphate group 5'. So that's an unusual linkage, and that unusual linkage protects the 5' end of that messenger RNA from degradation by exonucleases. It's also involved in translational initiation. So we have this 5' cap being protected. Okay, so it's going to be bound almost to the RBP1, the carboxy terminal domain of RBP1. And later on, when we look at translation, it will also be involved in translational initiation. Now the poly A tail we have gets formed because we have these polyadenylation factors, again, going to be bound to the carboxy terminal domain of RBP1. And as soon as this cleavage signal site here, this TTATTT, gets transcribed, so as soon as that sequence is in the messenger RNA, that attracts these polyadenylation factors. There's an endonuclease that will cut the messenger RNA, and then there's a polyadenylate polymerase that will add AAAAs to the end of it. Okay, and that is the end of transcription, and we have a mature messenger RNA. What we haven't gone into yet between the production of the GCAP and the production of the poly A tail and the release of the messenger RNA from the RNA polymerase is we have splicing. Okay, splicing happens before the addition of the poly A tail, but it's the most complex, and so I'm going to leave it to the end here. So, RNA splicing, removing introns from RNA transcript and splicing together the adjacent exons. And so what we're looking at is a situation in which we are removing these sequences here, introns, 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 and we are splicing together exons, exons, exons. The interesting thing is the average intron size, or the intron sizes, can range from 50 base pairs to 800,000 base pairs. The average exon size is only about 150 base pairs. So these introns are way, way bigger than the exons. Now, why has evolution retained introns? We'll get into that a little bit later. And you can see as we look at more and more complex organisms, right, here we have Homo sapiens, mouse, here we have Drosophila melanogaster, here we have our single-celled eukaryote, yeast cells. And the more complex we get, the more that introns, the higher the average number of introns per gene is. All right, there's 3 classes of RNA splicing. 2 of them are very rare. They're not used very much, only in very specific organelles or organisms. And that's group 1 introns and group 2 introns. And then there is the more common, the most common, is the spliceosomes. So let's take a look at these group 1 and group 2 introns that exist in just a few organelles and a few organisms. All introns, whether they require a spliceosome or are self-splicing, they all require a specific sequence that demarcates the 5' splice site, called the splice donor sometimes, and the 3' splice site, so the 3' end of the intron, and that's called the acceptor. And then all of them here have this branch site. Now, in group 1 introns, the branch site is a little bit different, and we'll look at why that's the case in just a moment. But for group 2 and for spliceosomes, this branch site is going to include an important A here, and this A is going to be important for the enzymatic, the reaction that's going to take place that eventually splices out this intron and splices together the 2 exons. All right, so let's look at the 2 self-splicing introns. The first one is group 1 introns. And now what group 1 introns are is they're introns that have a very specific sequence of RNA that allows them to fold in a way in which a free-floating G, right? This is a guanosine nucleotide here, is basically held into a pocket, like the active site of an enzyme. And in fact, this intron is doing that. It's acting exactly like an intron. And so it's going to be this hydroxyl group, the 3' hydroxyl group of this free-floating G nucleotide is going to attack the phosphodiester bond between the last base of the exon and the first base of the intron. That breaks the bond there. Okay, so now the intron here is the exon has been broken, the link between the intron and the exon. That leaves our 3' hydroxyl group at the end of the exon to make our second transesterification reaction, in which that hydroxyl group is now going to, in the same sort of reaction, attack the phosphodiester bond between the last nucleotide of the intron and the first nucleotide of the next exon. And so by the end of this, what we have is our intron and the spliced together exons. This intron never leaves the nucleus, and it will eventually be degraded, and now our exons are seamlessly spliced together. Let's take a look at that reaction, and I want you to be able to understand and be able to determine what is the nucleophile and what is the electrophile of this reaction, what's being attacked and what is doing the attacking. Now the attacker here is that 3' hydroxyl group from that free-floating guanosine that attacks the phosphate of the phosphodiester bond between the last exon and the first intron base. And so when that happens, then that covalent bond is going to be transferred from between the intron and exon to the intron connected to that guanosine, freeing up the 3' hydroxyl group of the exon. And that's going to be involved in the next transesterification reaction, right? The second one. Now, Group II introns are different than Group I introns in that they rely not on a free-floating guanosine nucleotide being held into a pocket. Now instead, that hydroxyl group is coming from an adenine that is within the intron itself. And because that adenine is already involved in a phosphodiester bond, we can't use its 3' hydroxyl group. But fortunately, since it's RNA, it still has a 2' hydroxyl group. And so when I ask which hydroxyl group of the branch point A is the nucleophile, that's it. It's got to be the 2' hydroxyl group. So that A there, the 2' hydroxyl group, is going to be the nucleophile. It's going to hit that phosphodiester bond, okay, breaking it. And now that's going to form a lariat structure, right? This structure here. The 3' end of that exon now is going to be the nucleophile that attacks the phosphodiester bond between the end of the intron and the beginning of the next exon, releasing that lariat and leaving the two adjacent exons spliced together. So Group I and Group II introns are similar in that they are both self-splicing. The introns themselves are acting like an enzyme. Okay, so they are ribozymes. The difference is that Group I requires that free-floating guanosine nucleotide, and Group II requires that A within the intron. In that respect, how it requires an A that is within the intron as the first nucleophile, that is how it's going to be similar to spliceosomes, and we'll get to that in a moment. But because Group I and Group II introns act as enzymes, they need to fold in a very specific way, and so the sequence of the introns matter. They have to have a specific sequence to fold in a specific way to have this enzymatic ability. Now, let's take a look at the more common way in which introns are spliced out in most eukaryotic organisms, and that's via something called the spliceosome. Now, the spliceosome is dependent upon 1, 2, 3, 4, 5 SNRPs, SnRNPs, U1, U2, U4, U5, and U6. Each one of these SNRPs is made up of a protein and a small nuclear RNA, or snRNA. And the RNA is going to be the guide and the catalyst. Okay, so the RNA actually, again, in this form of splicing, we're relying on RNAs as the enzyme. Now, the intron isn't going to be the enzyme. It's going to be the RNA within these SNRPs. The protein is just there to stabilize the RNA structure. So if we take a look at the RNAs that are involved in these SNRPs, we'll see how their sequence is important. U1, the snRNA of U1, is reverse complementary to the 5' splice site. And the RNA of the U2 is reverse complementary to what's called a branch site of the intron. And now that branch site includes the A, whose 2' hydroxyl group is going to be the nucleophile of that first transesterification reaction. So very similar to Group II introns. And notice that the way in which the sequence binds to the sequence here in the intron, it forces that A to not bind. It forces it to sort of bubble out. And that's what's going to make it the nucleophile. And then, of course, here, this sequence is important. This AG here to GU is going to mark the beginning of the exon and the end of the intron. Now, notice also that these RNAs that are involved in the SNRPs also are reverse complementary to other SnRNAs. So, for example, the SnRNA of U4 hybridizes with the SnRNA of U6. And the SnRNA of U6 hybridizes with the SnRNA of U2. And so all of these reverse complementarity aspects here between the SnRNAs and the intron and the sRNAs amongst themselves allows for this complex spliceosome to form and catalyze the splicing out of the intron and the splicing together of the exons. Let's see how this occurs. So the assembly. So first, U1 has the snRNA that is reverse complementary to the 5' splice site, binds there. U2 has an snRNA that is reverse complementary to the branch site, so it binds there. And then that attracts the U4, U5, U6 Tri-SnRNP complex that binds. And so now we've got this full spliceosome, but until U4 leaves, it's inactive. There's then a shift where U2 and U6 bind to one another, the snRNAs shown here. So there's the U6, here's the U2. That then positions the U2 snRNA right at the perfect place such that that A pops out and positions it so that it breaks the phosphodiester bond between the upstream exon and the beginning of the intron. That break then allows for the 3' hydroxyl group of the end of exon 1 to then attack the phosphodiester bond, keeping the end of the intron and the beginning of the next exon. And so there is our lariat form, right? Very similar to the group 2 self-splicing introns. And we get the 2 exons seamlessly spliced together. So the spliceosome also relies on 2 transesterification reactions. This is all coordinated by the carboxy terminal domain of RBP1 subunit of RNA polymerase II. And here we have elements of the spliceosome that basically sit here on that carboxy terminal domain and wait for a 5' splice site to emerge, and then bang, it jumps onto it. And then it just sits there and waits until the 3' end of that splice site comes out. Bang! It binds onto that. And then it excises that intron and puts the 2 exons together. So you'll never have a situation where more than one intron is exposed at the same time under normal circumstances. There is circumstances in which introns and exons will both be spliced out. We're going to go to that in a second. But the normal situation is that as an intron emerges, it immediately is spliced before the next one emerges. And so this is how a cell can maintain the accuracy of splicing. So the next video, we're going to talk about the alternative splicing of RNA. Depending upon what cell type we're in, we can splice RNA in different ways to generate different protein products.